Hi everyone! Today we're going to discuss a major drug class called benzodiazepines. Benzodiazepines are used to treat many diseases, but are primarily used to treat anxiety disorders. According to the Diagnostic and Statistical Manual of Mental Disorders, the DSM-5, anxiety disorders include a variety of disorders, like panic disorder, which are spontaneous panic attacks, generalized anxiety, which is persistent, unrealistic worry about everyday things, and social phobia, which is extreme shyness and fear of being judged by others. Anxiety disorders are also closely related to other classes of obsessive-compulsive, PTSD, and depressive disorders. They have high comorbidities with these related diseases, so benzodiazepines have also been used for some of these diseases, but is predominantly used for anxiety. The global burden of anxiety disorders affects 272 million people in 2010, which is about 4% of the world's population. Although anxiety disorders do not directly cause death, it is the number 6 cause in the world of years lived with disability. Anxiety disorders cause 390 dailies per 100,000 people. Dailies are disability-adjusted life years and are used to estimate decline in quality of life. A study compiled findings from representative samples of the general population in Western countries to compare the prevalence of anxiety disorders in these countries. These 12-month prevalence rates, as seen on the y-axis, reflect survey respondents who have experienced an anxiety disorder in the last 12 months. The diversity in the findings may reflect the differences in case definition, diagnostic instruments, response rates, and sample composition. The most common subtypes were specific phobias and social phobias, while the least common subtype was obsessive-compulsive disorder. The 12-month prevalence ranges from 5.6% in Australia to 19.3% in the United Kingdom. Most of the information about mental illness has been collected in Western countries. The World Health Organization developed a worldwide mental health project with nationwide surveys that share common methodologies to facilitate comparisons. They found that the most common subtype in non-Western countries were, once again, specific phobias and social phobias, while the least common subtype were panic disorders and agoraphobia. The 12-month prevalence ranged from 2.7% in China to 11.2% in Lebanon. These prevalence rates are much lower than that of Western countries, which could reflect cultural differences like social stigma and a lack of trust in anonymous survey research. Clearly, anxiety disorders affects many people across the world, and benzodiazepines can be used to decrease neural activity that causes anxiety.
This is a three-dimensional representation of the GABA alpha receptor. The file was obtained from the protein databank and rendered in PyMol. It is colored by secondary structure, with alpha helices being teal and beta sheets being purple. It is an ion channel, which is a transmembrane protein and delivers signal across the membrane to cause activity in the cell. The receptor becomes active when GABA binds to the GABA alpha receptor. This triggers a chloride ion to travel down the channel and stimulate the production of anion conductase, which hyperpolarizes depolarized neurons. Through this action, GABA is able to calm down overactive neurons by making them temporarily inactive. Benzodiazepines act at the GABA alpha receptor. They bind to the GABA alpha receptor and cause more chloride ions to flow through the channel, causing more anion conductase to be produced and more neurons to hyperpolarize, resulting in less active neurons. The GABA alpha receptor is made up of 5 subunits, 2 of which are identical. There are several isoforms of the GABA alpha receptor, but this composition is the most common in humans. Function depends on the arrangement and conformation of these 5 subunits, and we will see how these are modulated by benzodiazepines. The GABA alpha receptor has 2 sites for the endogenous binding of GABA. When both of these sites are bound, the receptor will perform its action. The neurotransmitter GABA binds to an active site on the receptor. The receptor needs two GABA molecules to trigger its mechanism. When two GABA molecules bind, the ion channel contracts and then expands to allow a chloride ion to travel down the channel. Benzodiazepines bind to an allosteric site on the receptor, which does not trigger the same mechanism that binding to the active site does, but changes the conformation of the receptor to increase its affinity for GABA binding. By this action, benzodiazepines are known as positive allosteric modulators, because although they do not bind directly to the active site, they still bind in a fashion that increases the average time the receptor spends open. Benzodiazepines can bind in a variety of sites, and each binding site has a different effect on the conformational change of the receptor.
In the 1950s, chemists were searching for new mood-altering compounds. Leo Sternbach was one of them, and he began studying compounds he had helped synthesize as a graduate student. In 1954, while he was working at Hoffmann-La Roche Laboratories, he was able to derive 40 new compounds that were all pharmacologically inactive. He decided to treat one of those compounds with methylamine and then stored it away. Almost 2 years later, a lab assistant found the compound while cleaning out Sternbach's lab and submitted it for evaluation. This compound, chlordiazepoxide, had sedative, anxiolytic, and muscle relaxant properties. In 1960, it was approved by the FDA and sold by Hoffmann-La Roche under the name Librium. Chlordiazepoxide is most commonly administered orally. Because it is fairly lipophilic, it is absorbed quickly and reaches peak concentration levels 0.5 to 4 hours after administration. Benzodiazepines are highly protein-bound, but rapidly distribute to the central nervous system, where they cross the blood-brain barrier by passive diffusion. The rate of distribution correlates with lipophilicity of the specific benzodiazepines. The volume of distribution of chlordiazepoxide is 0.25 to 0.5 liters per kilogram, and its half-life ranges from 5 to 30 hours. In patients who are elderly or hepatically impaired, clearance is typically reduced and half-life is prolonged due to difficulties metabolizing the drug. Metabolism of chlordiazepoxide and other benzodiazepines is performed by liver enzymes. Chlordiazepoxide is transformed into 4 pharmacologically active metabolites: desmethylchlordiazepoxide, demoxepam, desmethyldiazepam, and oxazepam. These metabolites produce therapeutic effects similar to the parent drug and can be easily eliminated through the kidneys. It is important to note that the duration of action of chlordiazepoxide is determined by the half-life of its active metabolites rather than the parent compound itself. Chlordiazepoxide can undergo a number of metabolic reactions. Shown on the screen is the metabolic process it undergoes to form one of its active metabolites, oxazepam. Oxazepam was even patented and marketed as a drug itself in 1965. Chlordiazepoxide can also undergo phase 1 hydrolysis to form its active metabolite, demoxepam. If it were to undergo phase 1 hydrolysis and phase 1 reduction of N-oxide, then the active metabolite, desmethyldiazepam, would be formed. Many metabolic reaction pathways are possible.
Since the introduction of chlordiazepoxide, many benzodiazepines have been synthesized. Currently, there are 3 benzodiazepines on the World Health Organization's list of essential medicines: diazepam, lorazepam, and midazolam. Small structural differences between these drugs lead to variance in pharmacological properties such as potency, speed at which they are metabolized, as well as different side effect profiles. Consequently, some benzodiazepines are better suited to certain purposes than others. Diazepam was introduced shortly after chlordiazepoxide in 1963, and with its numerous pharmacological advancements quickly replaced chlordiazepoxide as the standard of care for future compounds to beat. It is used to treat anxiety, insomnia, panic attacks, and acute alcohol withdrawal, and has also been used as a sedative. In the United States, it was the highest-selling medication between 1968 and 1982. Diazepam is extremely lipophilic, which allows for quick absorption and distribution to the central nervous system. Peak concentrations occur approximately 1.3 hours after administration. Additionally, diazepam's active metabolites and low clearance rate cause it to have one of the longer half-lives among the benzodiazepines, 43 hours. Compared to chlordiazepoxide, diazepam is about 2.5 times more potent. The pKi of diazepam at the GABA alpha receptor is 7.8. However, due to the complex nature of the GABA alpha receptor, it is difficult to find precise pKi values of chlordiazepoxide and other benzodiazepines for comparison. After the introduction of Valium, people made many more benzodiazepines. SAR analysis demonstrates that certain structural alterations are especially useful in promoting receptor affinity. Demitra and Honch illuminate that the most important features for elevating receptor affinity are having an electron-withdrawing substituent at R7, a small electron-withdrawing group at R' in position 2', and N-methylation at R1. Common electron-withdrawing groups at R7 are chloride and nitro groups, while the most common substituents for R' at position 2 on the aryl ring are chlorine and fluorine. Lorazepam was brought to market in 1977. The addition of chloride groups at position 2 increased receptor affinity, and likely partially explains the tenfold difference in potency between diazepam and lorazepam. Lorazepam is less lipophilic and has slower absorbance, reaching peak concentrations 1.2 to 2.6 hours after administration. Lorazepam also has a shorter half-life, as the hydroxide group off carbon-3 experiences relatively rapid glucuronidation, leading to a dead metabolite that is readily eliminated from the kidneys. It is best used for sedation and a short-term treatment of anxiety, insomnia, and acute seizures. It is worth noting the resemblance diazepam and lorazepam bear to two of chlordiazepoxide's active metabolites, desmethyldiazepam and oxazepam respectively. Clearly, the development of some of the most successful later-generation benzodiazepines were based largely on the structure of chlordiazepoxide's active metabolites. The final benzodiazepine on the World Health Organization's list of essential medicines is midazolam. In 2010, midazolam became the most commonly used benzodiazepine in anesthetic medicine. What makes midazolam so special is that it is soluble in water. Due to its great solubility, it does not produce negative side effects such as venous irritation, phlebitis, and pain on injection that are common with less soluble benzodiazepines, such as diazepam. Midazolam is also more potent than diazepam. The pKi of triazolam, a benzodiazepine that is extremely similar to midazolam, is 9.0 at the GABA alpha receptor, while it is only 7.8 with diazepam. Midazolam has a rapid onset of action, reaching peak concentration levels 0.67 hours after administration, a high clearance rate at 6.6 milliliters per minute per kilogram, and a half-life of 1 hour, making it the ideal drug for when short-term effects are desired.
Benzodiazepines are well known for being abused in a recreational setting. Most abusers take benzodiazepines with other drugs, such as heroin or other opioids, cocaine, or amphetamines to enhance the effects of those drugs. While benzodiazepines are sometimes used alone, they have only a small abusive effect unless they are used in doses greater than the therapeutic range on the dose-response curve. When abused, however, benzodiazepines are extremely addictive and have a wide range of withdrawal symptoms that include depression, nausea, memory loss, hallucinations, and sensory sensitivity to light, noise, and touch. The abuse of benzodiazepines have also been proven to carry great fetal and neonatal risks. Benzodiazepines have been widely used over the last 50 years to treat anxiety disorders by reducing activity of neurons to have sedative effects. They are still effective and commonly prescribed, but medical professionals are moving towards SSRIs, or serotonin reuptake inhibitors, because benzodiazepines have a high danger of addictiveness and potential for abuse. However, pharmaceutical companies still use the pharmacophore of this revolutionary compound as a basis for new drugs. If you were listening, today we discussed 1. How benzodiazepines allosterically bind to the GABA alpha receptor to increase receptor affinity for the neurotransmitter GABA. 2. How the first benzodiazepine was discovered in a lab cleanup. 3. How further generations of benzodiazepines, the LAMs and the PAMs, were based on chlorodiazepoxide's metabolites and improved on its ADME properties.